Hello, I'm Stuart Tugendreich, Director of Product Management at Ingenuity Systems. Today I'm going to demonstrate how IPA can be used to develop and evaluate biomarkers. I'll do a case study that explores both microRNA and mRNA biomarkers using data I pulled from the literature on ovarian cancer and show off some of the quick and powerful tools IPA provides for your own biomarker workflows. Toward the end of the talk, I will show you a prototype tool we are working on for doing gene set enrichment analysis, or GSEA, that we have running in a beta version of IPA. You're welcome to try out the beta version. I'll show you how later. Ovarian cancer is one of the leading causes of cancer-related deaths for women in the U.S., with over 14,000 mortalities last year. There are several known risk factors, but there is not yet any clear-cut test that can predict risk in most people. Interestingly, recent evidence suggests that the most common and aggressive ovarian cancer, which is called serous carcinoma, may derive from cells from the fallopian tube epithelium rather than from the ovaries themselves. Ovarian cancer is tricky to diagnose because the patient's symptoms are usually relatively nonspecific and often don't manifest until fairly late in the course of the disease. It is difficult to cure because it often recurs after treatment. Researchers are striving to find better methods to diagnose ovarian cancer early, select the best therapy, and identify patients who need to be followed the most closely for recurrence. For the following demonstration, I pulled two papers from the literature. One from Patrice Morin's lab at the NIH and Johns Hopkins that focus on microRNA expression in ovarian cancer, and the other from Stephen Canistra's lab at Harvard, which sought to develop a classifier of high-grade ovarian cancer. The microRNA dataset derived from tumors of 34 patients and found 55 microRNAs perturbed at least twofold in at least 60% of the samples. The mRNA dataset measured mRNA expression of high-grade serous epithelial ovarian cancer, coincidentally also using 34 patients, but split into two classes, 17 with fast recurrence, those who had a median survival of 33 months, and 17 with slow recurrence, those who had not yet reached their median survival at the 47-month point. I found that there were 348 genes that had a fold change greater than 1.5 and a p-value less than 0.05 between the fast and slow recurrence groups, and that's what we'll be using in the analysis you'll see today. So let's first focus on the microRNA part of the workflow. For this, I'll be using a new tool in IPA called the microRNA target filter. This slide conceptually explains how that filter works. So in step one, you upload either a list of microRNAs or microRNAs with expression data, and IPA automatically returns a list of predicted and or validated mRNA targets for those microRNAs. In step three, you can filter the list with this new tool by biological aspects of the mRNAs, for example, what pathways the mRNAs are involved in, what diseases they might be associated with, or by molecule type, for example, only looking at kinases. Since microRNAs generally affect the expression level of their targets, the tool enables you to upload mRNA gene expression for the validated and or predicted targets and filter for those targets that are downregulated when the targeting microRNA is upregulated and vice versa. At this point, as shown in steps 4 and 5, you can take the remaining microRNAs and or their targets and move them into other tools in IPA for further analysis. For example, you could run an analysis to determine significant associations with pathways or biological processes, create custom pathways, or further explore whether the molecules have been previously identified as known biomarkers in other contexts. This is all enabled by greater than 3 million biological findings, or scientific facts, structured and curated by us from the literature and from select biological databases. So now I will walk through how to take the microRNAs and their expression data from the first paper and upload it into the microRNA target filter in IPA. This shows the targeting table that automatically appears and demonstrates how the first microRNA, LET7, targets many mRNAs. The total of 48 microRNAs that were uploaded target over 14,000 mRNAs. The pathways that each mRNA participates in is automatically shown, and if desired, I could filter to retain only mRNAs that participate in cancer pathways, for example, or I could display additional columns to show each mRNA's relationship to disease, tissue or cell lines, and more. Instead, I'm going to upload expression data from the second paper to annotate each mRNA. That will result in a new column that can be subsequently filtered on. I will choose to retain only microRNA mRNA pairs that show opposite expression direction. That filter step reduces relevant targets from over 14,000 to only 255. Now I'm getting down to a more manageable set of genes for deeper biomarker analysis. For example, I can send the microRNAs, mRNAs, or both into IPA's analysis tools. Pathway analysis of the approximately 250 mRNAs left after filtering reveals that the IGF-1 signaling pathway is likely to be important in the more aggressive form of ovarian cancer. It is the top pathway amongst the almost 300 Ingenuity curated pathways that are tested in this analysis. IGF-1 signaling is involved in cell proliferation and cell survival. Overlaying the ovarian cancer mRNA gene expression onto the pathway reveals that many of the genes are affected. Red genes are upregulated and green are downregulated in fast versus slow recurrence. Now we can take the microRNAs that survive the expression filter and connect them to their mRNA targets, and it is apparent that many of them target the genes of the IGF-1 pathway. We can also use IPA to explore what biological processes and diseases are likely to be affected by the microRNAs and their targets. The top 3 biological processes and disease categories are all consistent with the ovarian cancer phenotype, with genital tumor being the most significant specific process in the reproductive disease category. To explore this set of genes more fully, I used IPA to create a network of the genital tumor genes and the microRNAs that target them. Many of the microRNAs are annotated with roles in cell growth and apoptosis, and 2 are known cervical cancer biomarkers. Furthermore, 3 of the mRNAs have been documented as ovarian cancer biomarkers. These are some of the findings we have curated from the literature that support each gene's association to genital tumors. It is easy to click through to the paper to dig deeper into the evidence. Another tool available in IPA is the biomarker filter. It takes any expression data set and allows you to add and filter the genes based on biomarker relevant criteria such as bodily fluids that the proteins have been detected in, or what kinds of biomarker applications are already known for each gene or protein. Here I have only retained genes that have a certain expression ratio in fast recurring ovarian cancer, are detectable in accessible biofluids, and have already been identified as biomarkers for some condition. In order to find specific biomarkers that are present in one condition and not in others, or are common across a set of conditions, you can use the biomarker comparison tool in IPA. In this case, I am looking for genes that are good biomarkers of fast recurrence ovarian cancer, but that are not differentially expressed in metastatic prostate cancer. I took the biomarker analysis that I just showed and simply added a biomarker analysis from prostate cancer. The tool enabled me to focus in on genes specific to the ovarian cancer. In a minute, I'm going to talk about our prototype GSEA tool that's available in our beta system, but before I do, let me briefly summarize what we've discussed so far. IPA offers a number of tools to support your biomarker discovery workflows. A new microRNA target filter to connect microRNAs to their targets and to find the key molecules that meet your criteria. I use it to find a set of microRNAs that target a set of mRNAs that exhibit opposite expression levels from the microRNAs in ovarian cancer tissue. So these are microRNAs that control mRNAs in the same tissue. We offer a biomarker filter to focus on molecules meeting biological criteria of your choice. I use it to focus on molecules that have been measured in readily accessible biofluids. This could aid in setting up a diagnostic assay. And we offer a biomarker comparison tool to do set operations to find the biomarkers that are in common or unique between groups of biological samples. I used it to exclude molecules that may also act as biomarkers of metastatic prostate cancer. Now let's take a brief look at the gene set enrichment analysis tool we have running on our beta system. GSEA is an approach developed by the Broad Institute that takes a set of gene expression profiles from 2 conditions and compares them against a library of gene sets. It looks for clustering of genes from a gene set at either the top or the bottom of the ranked data set. In this case, GSEA is looking for statistical enrichment for gene sets that are either relatively upregulated in fast or relatively upregulated in slow recurrence ovarian cancer. Our prototype GSEA tool employs several distinct Ingenuity proprietary gene sets libraries such as disease genes, physiological system development and functions, molecular and cellular functions, and transcription factors. We have provided a few simple settings for you to set up and run a GSEA analysis. The results are shown here are intriguing. Many of the top scoring functions are clearly related to phenotypes associated with aggressive forms of cancer. For example, attachment of fibroblasts, cell spreading of epithelial cell lines, the quantity of cell lines, such as endothelial cells, proliferation, etc. Please contact me directly if you would like to try this early prototype. In summary, IPA provides an industry-leading tool to identify and prioritize your biomarkers. The tools are integrated together and enable open-ended exploration as well as precise filtering for in silico validation of your data. IPA is powered by industry-leading biological contents to support your biomarker workflows, and the new microRNA target filter tool brings content and tools together for an unparalleled ability to prioritize and understand microRNAs and their targets. Thanks so much for your time today, and please start your free trial today at ingenuity.com. Thank you very much.